Cryobiology is the study of the effects of cold in biology, and it finds considerable applications in the field of transplantation medicine. So in this lecture I want to deal with the applications of cryobiology in transplantation. I've divided the lecture into three sections. First, I want to talk a little about fundamental research in cryobiology, to explain what we understand about the mechanisms of freezing injury and how cryoprotective agents work. Then I'll talk a little about cell preservation and tissue preservation. And finally, I want to deal with organ storage. Now, cooling has been used to preserve the survival of isolated cells and tissues for very many years. And of course this depends upon the well-known effect of lowering temperature on chemical and physical reaction rates. It can be shown, for example, that the oxygen consumption of renal cortical slices decreases roughly exponentially with falling temperature, reaching less than 5% by 5 degrees centigrade. Similar curves can be shown for the requirements for other substrates. Clearly, it would be expected that one would get longer survival of isolated cells the lower the temperature can be reduced, but unfortunately, as soon as freezing occurs, cell survival with most cell types drops to very low levels. So let us first look at what happens when cells are frozen. Now, cells, of course, are very complicated systems, so we'll look first at the simplest possible model, which is sodium chloride solution. This graph shows the freezing point depression curve for sodium chloride. In this and in subsequent graphs I've expressed the concentration of sodium chloride in mole fraction terms, which is perhaps a little unfamiliar. It is, however, more applicable to cryobiology because, as I will explain, as freezing proceeds, one reaches very high solution strengths, and the conventional methods of expressing concentration are no longer strictly applicable. So to orientate yourselves, let me say that a mole fraction of 0.0027 is isotonic in the case of sodium chloride. Now, this graph, as I say, shows the freezing point depression of solutions of increasing strength of sodium chloride. But it's important to realize that this graph also shows what happens when a solution of a single strength, shall we say isotonic, is cooled to its freezing point and beyond. Because what happens is that when the freezing point is reached, ice separates, which is pure water, and the solute becomes concentrated in the remaining liquid phase. And as cooling proceeds, further ice separates so that the strength of the remaining solution follows precisely that curve. This is perhaps more clearly shown in this graph, where I have represented isotonic sodium chloride by 1 on the y-axis and have plotted the proportional increase in strength of sodium chloride as the temperature is reduced. And you can see that there is a 32-fold increase in the concentration of salt by the time the eutectic temperature, -21 degrees centigrade, is reached. Now, clearly this must have very dramatic effects on any cells that are also present in the system. So let us look at these very simple drawings which show diagrammatically what happens. Now, here we have sodium chloride solution. The dots represent solute molecules, and the circles represent the membranes of cells. There is an equal concentration of solutes on either side of the cell membrane before freezing starts. When freezing occurs, ice separates as pure water, pure solid water, so the solute molecules become more concentrated in the remaining liquid, and there is then an osmotic imbalance across the cell membrane. The response to this is, of course, that the cells shrink. They lose water to equalize the osmotic pressure across the cell membrane. So you see there is ice outside the cells, but no ice inside. Now, of course, the loss of water takes time, which is dependent on the water permeability of the particular cell in question, and it would theoretically be possible to cool so quickly that the water could not leave the cell fast enough to freeze outside, and intracellular freezing could occur. But we will come back to that question in a few minutes' time. Now, these diagrams are all very well, but let me just show you that this does actually happen. Here I have a graph prepared by my colleague Tony Woolgar showing the mass of cell water in red cells suspended in sodium chloride solutions of increasing strength. And you can see that the cells shrink and reach a minimum volume at around about 1800 milliosmoles. Now, what effect does this shrinkage have upon the cells? Well, there are many effects. One of them is that the cell membranes change their permeability characteristics. Here we see what happens to cell potassium in red cells suspended in sodium chloride solutions of increasing strength. And you can see at around about 1800 milliosmoles, the point where the minimum cell volume is reached, there is a loss of intracellular potassium. This also coincides with an increased loss of cells, hemolysis. Here we see that hemolysis starts at around 1400 milliosmoles and reaches around 50% by 4000 milliosmoles. So we know that salt concentration rises during freezing, but does this provide an adequate and complete explanation of freezing injury? Well, it doesn't. For one thing, the amount of cell destruction occurring during freezing is much greater at a given osmolality than it is during constant temperature exposure to that osmolality. In addition, one gets the same cell shrinkage and the same change in cation permeability whether the osmolality is increased with salt or with sucrose. But during freezing, there is very much less damage if it is done in the presence of sucrose than if it is done in the presence of sodium chloride. We have also found that red cells become permeable to sucrose when they are frozen, but not when they are merely exposed to high osmolality without freezing. So clearly there are additional factors at work in freezing. Well, what are these? Well, I think it is fairly obvious that the two factors which occur in freezing but do not occur in the simple hyperosmolar model system are firstly that when cells are frozen and then thawed, they are not only exposed to high osmolality, but they are also resuspended in isotonic solution when the ice melts. Secondly, of course, freezing and thawing involve temperature change, which the simple hyperosmotic model system does not. Well, many years ago, Dr. James Lovelock, working at Mill Hill, investigated the importance of these other two factors, redilution and temperature change. What he did was to take a red cell system and to suspend it in sodium chloride solutions of increasing strength, and then resuspend the cells in isotonic sodium chloride and observe the hemolysis produced. And you can see he obtained a sigmoid curve of this sort. The next experiment was to calculate the temperatures which produced the same sodium chloride concentrations, and then to freeze the cells to these temperatures in isotonic sodium chloride and then thaw them. And when he did this, he found that he got a very similar curve. Within experimental error, they were the same. So that redilution after hypertonic exposure had in fact duplicated the effect of freezing. Lovelock also investigated the effect of temperature change under constant hypertonic conditions. And when he suspended red cells in sodium chloride solutions between 0.01 and 0.03 mole fraction sodium chloride and then cooled them from 37 degrees to 0 degrees centigrade, that is without freezing, he obtained greatly increased hemolysis. So it was shown that cells exposed to hypertonic conditions and then subjected either to dilution into their original osmolality or to temperature change were damaged, and the addition of these two factors to the hyperosmotic effect was sufficient to explain freezing injury. Now, all these experiments were done with relatively slow cooling rates, and I mentioned initially that there is a possibility that intracellular freezing could occur when cooling is rapid. So let us look and see whether cooling rate does in fact have any effect on cell survival. This is a graph showing the survival of human red blood cells after cooling at rates between 1 and 10,000 degrees centigrade per minute. You can see that survival increases as the cooling rate is increased to 3,000 degrees per minute and then falls off. Now, clearly the salt effects which we have been talking about account for the reduction in survival to the left of the peak. And the reason that the peak actually occurs is that as the cooling rate is increased, there is progressively less and less time for these salt effects to take place. But when a cooling rate, in this case of 3,000 degrees per minute, has been reached, then survival falls off. And we might expect that this could be accounted for by intracellular freezing. Now, Mazur has done some very interesting work on this. First of all, he calculated the proportional reduction in volume of cells at different cooling rates depending upon the rate at which water could leave them. This graph shows his calculations for red blood cells. The measure V over VI is in fact the volume over the initial volume. In other words, it's a percentage of the initial volume of the cells. If the cells were able to remain in equilibrium, they would follow the dotted line. But obviously, as they are cooled more and more rapidly, and here we have graphs for 1, 5 and 10,000 degrees per minute, as the cooling rate is increased, so they are progressively less able to maintain thermodynamic equilibrium and in fact contain significant amounts of intracellular water and are therefore liable to freeze internally. Now, Mazur showed that several different cell types with known different permeabilities to water did in fact show different peak survivals at different cooling rates. The next graph shows the response to cooling rate for 4 different cell types. Yeast had a peak survival at 10 degrees per minute, and yeast is relatively impermeable to water. Red cells, on the other hand, which are highly permeable to water, had a peak survival at a high cooling rate. And hamster tissue culture cells, which are intermediate in water permeability, had an intermediate peak survival rate. I would like to point out also on this graph that mouse bone marrow cells, which are very typical a mammalian nucleated cell, in fact showed no peak at any cooling rate, and I will come back to that in a minute. But this work of Mazur's did supply an adequate explanation of the effect of cooling rate, which has in fact since been confirmed by direct electron microscopic studies. I want to show you here 2 electron micrographs of cells which have been cooled to liquid nitrogen temperatures, that is to minus 196 at different cooling rates, and have then been freeze substituted so that we can see where the ice was present. Here we have a cell which has been cooled rapidly. The white areas were zones previously filled by ice. You can just make out where the nucleus was, some mitochondria, but the cell is full of small ice crystals. On the other hand, when these cells were cooled at only 1 degree per minute, they became shrunken. You can see that the cytoplasm is pulled right down upon the nucleus. It has become very dense, and there is no sign of any intracellular freezing. So here we have a direct demonstration of the effect of cooling rate on on the location of ice. Now, cryobiology would in fact have relatively little application to transplantation if one was limited to cooling in the absence of cryoprotective agents. And in fact it was the accidental discovery by Chris Polge, Audrey Smith and Sir Alan Parkes in 1949 that glycerol would protect against freezing injury that has made it possible to develop cryopreservation techniques for use in transplantation. Now this, as I say, was an accidental discovery, but it is now reasonably well understood how this sort of cryoprotective agent works. I want to show you a graph of the freezing point depression of glycerol solutions. This is the same sort of graph as the one of sodium chloride, which I showed earlier. The principal difference being that the minimum freezing point obtainable with a glycerol water system, the so-called eutectic point, is a much lower temperature. Another very important difference between glycerol and sodium chloride is that cells will withstand exposure to very high concentrations of glycerol, whereas they will not stand exposure to very high concentrations of sodium chloride. Now, if we construct from that freezing point depression curve graphs similar to the proportional increase in salt concentration graph, which I showed you earlier, you will be able to see that glycerol has very profound effects in aqueous solutions that are cooled. On this graph I have also shown the sodium chloride, the isotonic sodium chloride line as a dotted line for comparison. Each of the other lines represents a glycerol solution. The concentrations are shown in mole fraction terms but correspond, roughly speaking, to 1, 2, 3 and 4 molar glycerol. As you can see, as the glycerol concentration is increased, freezing occurs at a progressively lower temperature, and the subsequent proportional increase in glycerol concentration is much less as the initial concentration is increased. Now, of course, the thing which is concentrating glycerol in this system is the separation of water as ice. And it follows that anything else which is present in the system cannot concentrate to a greater extent than the glycerol, because both are being concentrated by the same event, the formation of ice. So that if we consider a three-component system consisting of glycerol, water and sodium chloride, the sodium chloride cannot concentrate at the same steep rate that it did in the simple system with water, but must concentrate at a much slower rate. Here we see the mole fraction of sodium chloride plotted against temperature in solutions containing no glycerol or 0.5, 1 molar or 2 molar glycerol. And you can see that as the glycerol concentration is increased, so the rate of increase of sodium chloride concentration is also diminished. Now, also shown on this graph, and I've taken this again from data by Jim Lovelock, also shown on this graph are points which represent the mole fraction of sodium chloride, or the temperature at which 5% hemolysis was found when red cells were also included in the system. And the important point is that the same degree of hemolysis occurred at roughly the same sodium chloride concentration, but at different glycerol concentrations and at different temperatures in each case. And the higher the glycerol concentration, the lower the temperature at which the same degree of hemolysis was observed. Now, I want to show you a direct demonstration of the effect of glycerol on the freezing of red blood cells. This is a film that was made by Audrey Smith at Mill Hill a number of years ago and shows red cells on a freezing microscope preparation, both without and then with glycerol. Here you see red cells in saline on the microscope slide. The temperature is reduced and you see the ice crystals moving in from the left-hand side. And you can see that the cells are still present in the preparation. Now the temperature is allowed to increase and the ice melts, and you will see that as it melts all the cells disappear. They become hemolyzed and there are no intact red cells left. Now we see the same experiment repeated in the presence of glycerol. The first thing to notice is that the ice structure in the presence of glycerol is rather different. You'll see the ice crystals come in a moment as the temperature falls. And the liquid spaces between the ice crystals are rather larger than they were in the absence of glycerol. And the red cells can still all be seen clearly present in the preparation. You can see them moving around slightly, showing that there still is significant liquid phase present. During thawing, the liquid phase progressively increases in volume and the cells can be seen moving around before all the ice has gone. And finally, when all the ice does melt, you can see that there are large numbers of intact red cells still present. Now, glycerol, of course, is still very widely used as a cryoprotective agent, and there are other agents of the same type in common use, substances like dimethyl sulfoxide, dimethylacetamide and dimethylformamide. There is, however, another class of cryoprotective agents, the polymers like polyvinyl pyrrolidone and dextran, which were thought for many years to have a different mechanism of action. This thought arose because these compounds, having a high molecular weight, are present in relatively low molar concentrations when prepared in the standard sort of solutions in weight for volume terms. Now, it turns out that this is not correct. Some time ago, my colleague John Farrant showed that solutions of polyvinyl pyrrolidone do in fact produce profound depression of freezing point. Although there is little depression at low concentrations, the curve is markedly nonlinear, and at high concentrations there is a very profound effect. And of course, during freezing, as ice separates, very high concentrations are produced. So one can explain the cryoprotective effect of PVP on this explanation. There is, however, an additional factor with the polymers, and that is that when cells are thawed, they are, as I mentioned, relatively permeable to cations, and therefore the internal colloid is unopposed by external impermeable cation, and the cells tend to lyse. Now, in this graph I have shown an experiment by Tony Woolgar where red cells were exposed to strong salt solution and then rediluted into solutions of differing osmolality, either made up entirely of sodium chloride, and you can see that as the sodium chloride concentration is increased, so the survival rate improves. But this is compared with a solution containing isotonic sodium chloride and progressively increasing amounts of PVP. And you can see there is a marked improvement in survival. Now, this is explained by the fact that the PVP is impermeant to the thawed cells and therefore balances the internal colloid and prevents colloid osmotic hemolysis. Now, this has necessarily been a rather brief review of basic cryobiology, but I hope it will form an adequate background for discussion of some of the practical applications that I want now to deal with. The cryopreservation techniques for red cell storage that I've been describing find very useful application in clinical medicine. And we thought it useful, therefore, to come to the Northeast Metropolitan Blood Transfusion Centre at Brentwood, where Dr. John Blagden, the Deputy Director, will explain the system which is used here. Dr. Blagden.
Blood donations are collected in the normal way into standard packs. They're then spun to separate the plasma from the cells. Plasma is then removed and glycerol solution is added to give a final weight volume of 20%. They are then transferred into an aluminium canister ready for freezing. Freezing is carried out in liquid nitrogen. We make our own liquid nitrogen in the plant on the left. The storage of the units are in 4 nitrogen refrigerators supplied from a central reservoir with automatic filling and alarm systems. The units are frozen in liquid nitrogen to bring them down to minus 196 degrees centigrade. This takes about 9.5 minutes. They are then transferred from this vessel into one of the nitrogen refrigerators for storage until required. The lower units are stored to about this level in liquid nitrogen at a temperature of minus 196. The upper layer is in the vapor phase with a temperature of about minus 180 to a maximum of minus 160 at the upper level. They can be stored in this condition for about 2,000 years, theoretically. Before transfusion, the glycerol has to be removed. This is done by 2 methods. But first, the thawing is carried out in an agitating water bath at 42 degrees for 6.5 minutes. This is the continuous wash centrifuge method. After thawing, the red cells are transferred to a plastic pack. The wash solutions are held in cradles above the blood. They then flow through the tube into the centrifuge bowl. The bowl was developed by Latham from the cone fractionator in the United States. The inlet port, the cell suspension goes into the bowl and is held by the centrifugal force while the wash solution follows, washing the glycerol from the red cells. The exit takes the supernatant out into a pack in front of the machine. After washing, the red cells are siphoned off into the transfusion pack. For the manual wash procedure, the thawed red cells are transferred into a 5-tail pack. First line is used to transfer. This is sealed, and then the pack is centrifuged. The supernatant is then separated, and the first wash solution run through on the mechanical mixer. This is repeated for 2 further washes using 3 and 4. A final centrifugation and the final supernatant is then removed. The line is sealed and the pack is ready for transfusion. Using this procedure, one recovers 95 to 97% of the cells. On transfusion, the post-transfusion survival of the red cells at 24 hours is about 90%.
Red cell preservation is by far the most clinically important application of cryopreservation today, although techniques are currently being worked out for platelets and we hope also for granulocytes. There are two other applications of cryopreservation more closely allied to transplantation that I'd now like to talk about, and the first of these is bone marrow transplantation. It was found by Barnes and Loutet in the 1950s that the hematopoietic cells of the mouse could be preserved by a technique very similar to that now used for red cells. I have over here a graph which shows the survival of hematopoietic cells from the mouse at different cooling rates and in different concentrations of glycerol. Now you'll remember that earlier on I showed that there was no survival of mouse bone marrow cells at any cooling rate in the absence of a cryoprotective agent. That's this line here. But as the concentration of glycerol is increased, 0.4 molar, 0.8 molar, 1.2 molar, so increasing maximum survival rates are obtained, and the cooling rate giving the maximum survival rate becomes progressively slower. This, of course, is because glycerol is limiting the damaging effects to the left of the peak. And as more glycerol is present, so more of the area to the left of the peak is revealed and the maximum survival is obtained. In this case, at 1 degree per minute, in a concentration of 1.25 molar glycerol. Here we see the bone marrow cells actually being used in irradiated mice, and we have a comparison of the leukocyte count at various days post-irradiation, the transplant having been given immediately after the irradiation, and you see compared fresh marrow cells and an equal amount of frozen bone marrow cells. And you can see that there is no significant difference in survival between the two techniques. So the banking of bone marrow is a perfectly feasible technique, and there is no reason why this cannot be used in clinical medicine at the present time, where the proper clinical indications for marrow grafting exist. These are not many, but as you will know, there is quite a lot of interest in this at the present time, and bone marrow transplants are being done on an increasing scale. Techniques for preservation of skin and cornea were also worked out several years ago, and both of these can be used clinically. First, the skin or the cornea is soaked in a solution either of glycerol or dimethyl sulfoxide. There is some evidence that it may be better to use glycerol for the epithelial surface of the cornea and dimethyl sulfoxide for the endothelial surface. In each case, a concentration of approximately 1.2 molar is used. The tissue is then cooled slowly at 1 degree per minute or thereabouts, and may be stored for very long periods of time at liquid nitrogen temperatures, that's minus 196, or even at minus 80 if the storage period is not required to be much longer than a year. Both of these techniques have been used clinically, and again, where the clinical indications are appropriate, there is no reason at all why these techniques cannot be used on a much wider scale than they are at present. By far and away the most important organ that is clinically transplanted at the present time is, of course, the kidney. Now, the immediate requirements to make transplantation a possibility are only for relatively short-term preservation. Sometimes 4 or 5 hours is sufficient, but 12 or 24 is certainly enough to make the whole thing a practical proposition. However, there are very good reasons, largely stemming from immunological advances, why it would be an advantage to be able to store kidneys for a much longer period of time and set up true kidney banks. However, let us first look at the immediate problem of storing kidneys for up to 24 hours to make transplantation a possibility. Now, when transplantation first got underway, Roy Kahn and I investigated this problem using dogs, and we took a very simple approach, which was to remove dog kidneys, cool them in crushed ice to just above zero, and see how long they could be kept in that way and still support the life of a bilaterally nephrectomized dog after transplantation. And we found that up to 12 hours their function was perfectly adequate, although we sometimes got some acute tubular necrosis, and nonetheless the dogs all survived. But after longer than 12 hours, results became irreproducible, and eventually one got no survivors at 24 hours storage at all. Now, two very simple modifications to this technique have made it very much more successful. They were both quite empirically found. The first was to wash out the blood before commencing storage. Now, the most probable reason why this is advantageous is that red cells in the static microcirculation become depleted of ATP. And when this happens, the intracellular calcium, which is normally chelated by ATP, becomes bound to the cell membranes, which then become rigid. This graph shows some work by Weed, where he measured the pressure necessary to deform red cell membranes after the cells had been incubated for increasing lengths of time. And you can see that by 24 hours they had become really quite rigid. And when ATP was regenerated by adding adenosine, then the normal pliability of the red cell membrane returned. Quite obviously, rigid red cells of this sort would be very difficult to dislodge from the microcirculation. So washout is an advantage. The next empirical advance was the discovery that if the washout solution is rendered hyperosmolar, that results are very much better. Now, when kidneys were washed out with hyperosmolar solutions, and here we see lactated Ringer's solution rendered hyperosmolar with either glucose or sucrose, then the water content remains constant during storage, whereas in the absence of glucose or sucrose, the water content increases. In other words, the kidneys become edematous. Now, this is probably particularly important in the case of the endothelial cells. Here we have a measurement of endothelial cell thickness in normal kidneys and in ischemic kidneys, where you can see it has in fact doubled, and this will produce a profound effect on the vascular resistance of the kidney. In this experiment, mannitol was added to the washout solution, and you can see that this enabled the endothelial cell thickness to remain at its control value. And many different sorts of washout solution have in fact been used, and it was very difficult to be sure which was better because the experiments were all done in different laboratories and so on. So in our laboratory we did a comparison of a range of washout solutions which were modified each from the next in a systematic manner, and I have the results on these charts here. We've measured the function of the kidney after preservation by measuring in an in vitro system glucose uptake or sodium uptake. This is how controlled, freshly isolated kidneys perform. This is how kidneys removed from the animal and not washed out at all, just stored for 24 hours function, very little function. This is how kidneys function that have been washed out with a solution having the same composition as plasma, but the same osmolality, no added glucose, sucrose or mannitol. Again, rather poor function. However, when the osmolality is increased to 400 milliosmoles per kilogram by the addition of glucose, very much improved function is obtained. In this solution, the same osmolality was used, but the sodium concentration was reduced and the magnesium concentration was increased. This made no significant effect. In this solution, the sodium concentration was reduced and the potassium concentration increased. And you can see that this significantly depressed function. These two experiments were with continuous perfusion with a solution containing dextran. That had an osmolality of 300 milliosmoles per kilogram, and that one 400 milliosmoles per kilogram by adding additional glucose. You can see that these results are by no means as good as the simple washout solution, but that increasing the osmolality did improve function over the one of normal osmolality. This chart shows the comparable experiments, but using sodium uptake as a measurement of viability, and you can see the pattern is much the same. So our conclusion from these experiments is that a washout solution of high osmolality is certainly beneficial, but that other modifications that have been suggested by others, such as increasing the magnesium and the potassium concentration, are not in fact beneficial. I would now show you the actual composition of our solution. You can see the ionic composition is similar to that of plasma, but we have added sufficient glucose, 107 millimoles, to raise the osmolality to 400 milliosmoles per kilogram. We also have heparin and a vasodilator procaine present, and the solution is buffered with HEPEs. Now, these experiments in the laboratory have all been done with kidneys that were removed from animals in good health. There was no period of warm ischemic injury before preservation started. And of course, although this is an ideal situation, it is not one that is commonly encountered in transplantation practice, in this country at least, where most kidneys have suffered some warm ischemic injury before they can be removed. In these circumstances, washout preservation may not be the best technique. Some years ago, Bob Johnson in Newcastle made direct comparison of washout with Collins solution, which is a high osmolality washout solution, and continuous perfusion with PPF, plasma protein fraction, which is a crudely fractionated albumin solution. And you can see that as the warm ischemia time is increased, so the survival decreases, but at all warm ischemia times, above very brief ones, continuous perfusion gave better preservation. Now, continuous perfusion probably is the method of choice in Scandinavia, certainly. In some parts of the continent it is used a great deal, and in North America it is widely used. But in this country it has not been so popular, I think largely because of the complexity and the expense of the machinery. I have over here a diagram of the equipment which we use in our laboratory, and you can see it is a good deal more complicated than just putting a kidney in a thermos flask. The kidney is suspended, in this case, from a weight transducer so that we can continuously monitor edema. The venous effluent falls into a reservoir where the pH is maintained by passing 5% CO2 in air over the perfusate. It is then pumped through a submicron filter, a bubble trap, to an arterial cannula where the pressure is monitored. And the pressure is used to feed a process controller which regulates the pump speed so that we maintain a constant perfusion pressure of 40 millimeters of mercury. And both the weight and the pressure are continuously recorded. The perfusion equipment itself is enclosed in a temperature-controlled cabinet at 5 degrees centigrade. Now this, as I say, is a piece of equipment that was built for experimental use, but comparable apparatus is in use clinically. To show you some organ perfusion equipment in actual clinical use, we have come to St. Bartholomew's Hospital, where the Institute of Urology Gambro perfusion machine will be demonstrated by Mr. O'Donoghue. You can probably hear the machine operating in the background. Well, I'm afraid we don't have a kidney in it today, but Mr. O'Donoghue will now show you how it operates. Thank you.
This is the Gambro kidney preservation machine, which we have been using at the Institute of Urology and at St. Bartholomew's Hospital for some time now. In this particular view, the front cabinet of the machine has been opened so that you can see the perfusion circuit. Here we have two separate organ containers so that two kidneys can be perfused at the same time. Here one sees the pump by which the perfusate is pumped around here and up through a filter, then to a bubble trap, and then around to the organ container, and so through the kidney. The perfusate is continuously recirculated through each kidney at a low temperature, the temperature range being 5 to 9 degrees centigrade. During perfusion, perfusate pressure may be monitored here, and perfusate flow from the control panel at the side. The original machine has proved effective in use, but is rather bulky, and so is difficult to transport between transplant centers. This is the latest Gambro machine, which you can see is very much smaller and more compact, and so is much more readily transportable, and I think will be of much greater use in clinical transplantation.
In continuous perfusion techniques, it is very important to get the right perfusate. I showed earlier on some experiments in which a dextran solution was used, and as you saw, the results were not as good as our best washout method. Now, the machines in current use employ either cryoprecipitated plasma, which is complex and expensive to produce, or more commonly, highly purified albumin solution, and this is the one which is usually used in the Gambro machine. I think that the reason that continuous perfusion gives better results with ischemic kidneys than washout is again to do with the maintenance of a patent vascular system. Clearly, if the vascular system is not open at the time of transplantation, then it will be impossible for the recipient blood to flow through the kidney, and even if the kidney tissue itself is well preserved, it will rapidly die if it doesn't have a blood supply. So continuous perfusion can be used for preservation under optimal conditions for 2, perhaps 3 days. Now, the extension of the preservation time from 1 day to 3 days is occasionally useful, but to take advantage of immunological techniques that are coming along, it will be necessary to have very much longer preservation than that. And I cannot see how continuous perfusion methods could ever provide an adequate solution to this problem. They would tie up too many machines, one machine to each kidney, and it would never be possible to have a large bank using this sort of technology. Therefore, we are very actively investigating the possibility of using true cryopreservation techniques for kidney storage. The first problem one comes up against in trying to apply these methods to renal preservation is the simple one of trying to get the cryoprotective agent actually into all the cells of the organ, and then to remove it subsequently without damage, that is, even without going to sub-zero temperatures. We've studied 3 cryoprotective agents so far, glycerol, dimethyl sulfoxide, and ethane diol. And from our studies it appears that glycerol is the least toxic. I have here 2 charts which show the effect on vascular resistance and on weight gain, that is edema, of perfusion with 2 molar glycerol at 2 temperatures, at 37 degrees and at 5 degrees. You can see that when the cryoprotective agent is introduced into the perfusion circuit, the vascular resistance decreases. This is due to osmotic shrinkage of the endothelial cells, widening the capillary channels, and thereafter the vascular resistance stays constant. Another problem that one is worried about in organ perfusion is the development of edema, and you can see that when kidneys are perfused with 2 molar glycerol at 37 degrees, there is a large increase in weight gross edema develops, whereas when they are perfused at 5 degrees, the weight remains just below the starting weight, and there is no progressive edema. So we have studied 2 molar glycerol perfusion of isolated kidneys and are able to get it in all right. The next problem, however, is to get it out again, and here one comes up against much greater difficulty. This graph shows the vascular resistance of a kidney in an experiment where 2 molar glycerol was introduced, where it is shown, and perfused for just under 100 minutes, and then a change was made instantly to a perfusate without glycerol. And you can see there is a vast increase in vascular resistance. In fact, virtually no perfusate was passing through the kidney, indicating gross vascular damage. Now, we thought we might be able to improve the situation by including in the perfusate a compound which would not penetrate the cells and would therefore provide an osmotic balance to the intracellular glycerol while it was being removed. The next graph shows what happened when we added 100 millimolar mannitol and repeated the experiment. And you can see that the increase in vascular resistance was very much less, but still, at the end of the experiment, we had roughly 5 times the initial vascular resistance, and there must have been considerable damage to the microcirculation. It seemed logical that changing the glycerol concentration more slowly might produce less damage. We therefore repeated the experiment and changed the glycerol concentration at 1% per minute, as shown here, still keeping 100 millimolar mannitol in the perfusate. And you can see this did indeed result in a considerable improvement. And when the rate of glycerolization and deglycerolization was slowed still further, we got an even better result. Now, we are currently investigating the viability of kidneys, glycerolized and deglycerolized in this fashion, both by an in vitro technique and by transplantation. And when this work is complete, we shall go on to cool as the glycerol concentration is increased. This is work following on an original suggestion by John Farrant, who showed that smooth muscle tissue would survive cooling to very low temperatures if the glycerol concentration, or in his case the dimethyl sulfoxide concentration, was increased during cooling and decreased during warming, in order to prevent the occurrence of freezing at all. Now, to apply this sort of idea to a whole organ is some engineering undertaking, and I want to conclude this lecture by visiting my lab at Northwick Park, where you will see such an experiment in progress. Now, this is the equipment which we are using for the experiments in which rabbit kidneys are being perfused with increasing concentrations of glycerol and are being cooled at the same time. The perfusate is prepared in this thermostatically controlled mixing chamber and is continuously circulated by the pump here through a refractometer which measures the glycerol concentration continuously and around through the piping back into the reservoir. A mixture of 5% CO2 and oxygen is also passed through the reservoir continuously to maintain a constant pH of 7.4. Some of the perfusate is aspirated at this point here by the perfusion pump at the back of the equipment, which passes it through a submicron filter and down through a heat exchanger to the arterial cannula, which is tied into the renal artery of a rabbit kidney. The venous effluent passes into the small collecting chamber below and then is taken by this pump and returned back to the mixing chamber. Now, the kidney is suspended from a weight transducer. It's actually mounted just below, so that we can monitor the kidney weight continuously. And the perfusate pressure is monitored by a connection to the cannula, which leads to a pressure transducer here. The perfusate temperature is monitored by a thermocouple in the perfusate tubing just at that point here. Now these three variables are continuously recorded on a 3-channel chart recorder, and in addition we make intermittent measurements of perfusate flow rate so that we can calculate the vascular resistance of the kidney. Now the glycerol concentration and the temperature of the perfusate are continuously controlled by the equipment in this rack. Now this unit contains the programmers, and the temperature program is continuously displayed on this meter, and the glycerol concentration program on this meter. The programs are actually generated by a linear ramp generator and by a card reader which can produce curved programs. Now in this unit we have displayed the actual measured temperature and the actual measured glycerol concentration in the perfusate, so that we have available the desired temperature, glycerol concentration and the measured glycerol concentration. Now, these data are fed into two process controllers here and here, and the outputs of these controllers operate the perfusion equipment to produce the desired temperature and glycerol concentration throughout the experiment. The concentration controller operates this pump, which takes fluid from this reservoir and injects it into the mixing chamber in the required amounts to produce the necessary concentration at that time. During glycerolization, the reservoir contains 40% glycerol in perfusate, and during deglycerolization it contains plain perfusate with no glycerol. The temperature controller controls a refrigeration unit which alters the temperature of coolant flowing through the heat exchanger here, and thereby controls the temperature of perfusate flowing through the kidney. Now, the experiment that is going on at the present time is one of a series which are designed to determine the optimum rate of addition and rate of removal of glycerol from rabbit kidneys at a constant temperature of 5 degrees centigrade. When these experiments are complete, we shall then go on to add cooling to the program. Glycerol concentration will be increased during cooling. The kidneys will then be stored at a low sub-zero temperature. And during rewarming, the glycerol concentration will be reduced again on this equipment. The ultimate object, of course, being to develop a method for the long-term storage of kidneys for transplantation.